Hello and welcome to Amines 4. So this lesson's called Amines 4, but in reality it's really more about amides, which are a kind of related and easy to confuse compound, but they are not the same. So the focus of this lesson is synthesis of amides, which we know a little bit about already if we've studied acylation, and the hydrolysis of amides. Right then, let's crack on. Pause the video. Try and answer these questions on a whiteboard or on a scrap of paper as usual. And then once you've had a chance, you can see the solutions. Right then, so balanced simple equations, okay. Sort of borderline tougher acid-base equations involving amines. All right, there's not an awful lot for me to comment on apart from the fact that some of the acids react with more than 1 mole of an amine, for example, the sulfuric and the phosphoric. Just the usual comment applies, which is if you've got separate ions as your products, that's okay, as long as it's correct. If you've got separate ions instead of a complete formula, that's all right. Okay, let's move on. The rank order of the amines that you were asked about looks like that. Okay, a little comment on this. You might well have got ethylamine, methylamine, and ammonia correct, okay? But you might have been struggling a little bit with diethylamine, okay? That's a secondary amine, so it should be higher up. Actually, I tell a lie, that's not what I meant at all. What I meant was you might have got this one, and this one correct, but not been sure about ethylamine. Apologies, okay? So the idea is you might not have known how to distinguish between the two primary amines. And the idea is, remember, we're thinking about that positive inductive effect. And although they're both primary amines, ethylamine has a longer carbon chain kind of pointing towards that nitrogen. So you're going to get a greater positive inductive effect. Okay. So here are the facts about amides. You should have seen these already if you've studied acylation, but it's worth reminding you. To spot an amide, you look for a carbonyl group, C double bond O, adjacent to a nitrogen-hydrogen. That is what an amide is, the whole thing that's there, CONH, okay? All amides contain that. They tend to be synthesized from amines and carbonyl compounds, although there are a number of different routes you can get to them with, okay? And you find them in a number of different compounds. Amides is the simplest one. N-substituted amides is another one. But then you also find them in biomolecules such as peptides, which are, essentially what are then folded and formed into proteins in biochemistry, and synthetic polyamides as well. We're going to cover polyamides in the next lesson, but it's just sort of for information. Okay, so if you've paused the video and made your notes as appropriate, let's keep going. So here's our first example of how to synthesize an amide. You can react together carboxylic acids and amines, and in a condensation reaction, water will be produced as a byproduct, and you will form an amide. So what I want you to do, rather than just going straight into a task, we'll do one afterwards, but I also want you to have a go at this, right? You should really pause the video, try and predict how to draw the structure of the amide that you get from this reaction and the byproduct. So if you've had a go at that, what we should be looking at is that. Okay, just to point out, that R and that R are the same. That R and that R are the same, okay? And that's important because obviously whatever's attached to the carbonyl compound, in this case the carboxylic acid, has to stay attached to that carbon, and whatever's attached to the nitrogen in the amine has to stay attached to that nitrogen, okay? Water is then, removed as a byproduct, all right? So that's what happens there. We don't need a mechanism for this, all right? We just do equations and sort of predicting, the formula or the name of products. We can't name this one because obviously it is not a sort of specific amide. It's a general structure. And remember, the R that's on the left could be CH3, or it could be CH2CH3. You know, it could be propyl, butyl, whatever. And the same goes for the one on the right. Right then, this time, you're going to do it all. So what you need to do is pause the video and read through this task, okay? My suggestion is to draw the structures of these specific carboxylic acids and amines facing one another, like this generic structure. Realize and deduce that you need to basically subtract the components of a water molecule from those two structures combined and link them up and replicate the structures that were in the reactants, okay? So pause the video and have a go at this, get as far as you can. Right then, so no solutions for this just yet because I want to talk through it a little bit, all right? There's your ethanoic acid. There's your ethylamine. Okay, and this is what I meant by face the two molecules against each other. Quick reminder that you're going to get rid of water from here in order to link the two molecules as an amide. And I'm going to point out that I would draw it in this order. I know that an amide is going to be formed. So the first thing I'm going to draw is my amide linkage, the CONH. And then I'm going to reference the reactants and I'm going to track to the left, which I know has an additional methyl group on it. And then I'm going to track to the right, which I know has an ethyl group. And that is my structure. I'm also going to point out that water has been released as a byproduct because it's a condensation reaction. The only other thing that we could do, although I don't think I asked you to do this specifically, is name this amide, okay? This is a specific kind of amide called an N-substituted amide. So the name follows the pattern N, and then whichever alkyl group is attached to the nitrogen goes here. So that's ethyl. And then whatever is attached to the carbonyl, or whatever, the carbonyl bit inclusive goes at the end. And that's the bit that takes the form something anamide. In this case, ethanamide. Just in case that wasn't clearly explained enough, which it might well not have been. Okay. Use a bit of color coding to explain. That part of the name corresponds to that section of the molecule. That part of the name corresponds to that section of the molecule. Okay. Again, this is a bit like naming esters, where basically you've got, two components to the name, one of which is derived from one of the reactants, the amine, and the other one is derived from the other part of the reactants, which is the carboxylic acid. Let's keep going. Same sort of idea. Propanoic acid and methylamine. Just preference that I do them in, in this particular order. You don't have to do them the same way as me. But again, I'm going to start in the middle and then work outwards. Okay, so from the carbonyl, I'm going to go back and add the stuff that was in the carboxylic acid. Okay. Then from the nitrogen, I'm going to add the stuff that was in the amine. And then I'm going to put water. So this is still an N-substituted amide. This time it's N-methyl propanamide. Okay. Next, naming is going to go out of the window soon because we're going to involve some aromatics. We don't need to be able to name these. All right. So this time we've got benzene carboxylic acid. This is something that the AQA have a tendency to do, where they like to incorporate aromatic rings into synthesis questions. even though in a lot of the cases, there isn't actually any of the aromatic ring involved in the reaction. So in this case, basically, there's a carboxyl group attached to a benzene ring. The benzene ring is going to be largely irrelevant. It's going to appear in the structure of the amide, but it's not actually going to do anything in terms of the reaction. The reaction's still happening between the carboxyl group and the amino group. So there we go. Same sort of logic. I'm still going to start at the amide linkage. I'll do this bit first this time, which is the methyl group, and then I'm going to do an elongated bond to make sure that I've got plenty of space to draw my benzene ring. Okay? So there you go. Looks dead complicated, but it isn't really. Okay? I guess you'd call this, I'm guessing really, but N-methyl, I'm not going to commit it to writing, but N-methyl benzanamide, something along those lines. Okay? So let's keep going. Ethanoic acid and phenylamine, we've got the same sort of idea here where I've thrown in an aromatic ring. But it's not actually going to be important because it's really only where the amino group and the carboxyl group is that there's a reaction. CONH, CH3, benzene ring. I feel like I messed up the name of the previous one, so I'm not going to commit to a name here, okay? Because again, it goes beyond the scope of the A-level stuff, all right? Obviously, it would end ethanamide, N-phenyl ethanamide, I suppose, okay? Right then, let's move on. So just to comment on the biomolecules that we call peptides or, eventually polypeptides. So these are a special kind of amide, and they're special because they're made from condensation reactions of amino acids. But the thing that's particularly special about amino acids is that they can self-polymerize. What I mean by that is, normally, when you're looking at an amide, right, you have to react two different kinds of molecules together. We've just looked at carboxylic acids and amines. And in this case, you're doing that. You're reacting a carboxylic acid and an amine, but actually, it's entirely possible to react many molecules of the same amino acid together, right? Because each amino acid contains an amino group and each amino acid contains a carboxyl group. So what we're getting at is, if you take, for example, this amino acid, which we don't know which one it is because it's just got a general structure, and you times it by 2, in other words, if there are two of these things next to each other, you can get a condensation reaction and an amide can form. One end of this molecule can react with the other end of a different molecule, of an adjacent molecule. Okay? And if that happens, the structure is called a dipeptide, and it looks like this. So just to be clear, right, we were saying if you multiply that by 2, if we've got two of those next to each other, then the carboxyl group on one end of one molecule and the amino group on the other end of another molecule, an adjacent molecule, can react together, form water as a byproduct, and you get an amide, a special kind of amide called a dipeptide. Now, in this case, our dipeptide has 2 of the same amino acid in sequence. And actually, it's a really important function of biology that, what's attached in the R position, the variation of that is what actually sort of creates the molecular architecture of proteins, well, of polypeptides and then of proteins. Okay, it's not an accident, basically. Organisms code in a particular way to create polypeptides that have a special sequence where there's something here, then there's something there, and then it carries on and on and on in a polypeptide. Okay, and that's very important because it basically causes polypeptides to fold in a particular way in 3-dimensional space, which leads to the formation of proteins. So it's a slight tangent, but it's really quite a significant thing. When it comes down to the sort of function of it, I'm now going to ask you to use your data book to draw the following peptides. So just to be clear, this bit tells you how many units there are, okay? And the bit to the left of that tells you in what order they're going to connect. And all of these amino acids are in your data book, so you don't have to learn their structures, obviously, but you do need to be able to put them together, okay? So we've got a dipeptide, which is 2 units, alanine, then serine, in that order. And then 2 tripeptides, where it's the same 3 amino acids, but they are in a different sequence. And then finally a dipeptide with some fairly complex looking amino acids in, but also the additional complication that it's at pH 7. If you don't feel like you get all of this right, don't worry, but pause the video and have a go before we have a look at the solutions. Okay, so you should have some answers. Might not be all of them, but you should have some answers. Let's have a look. There's the alanine serine dipeptide. Okay, the bit on the left is the alanine, the bit on the right is the serine. Crucially, there is an amide linkage in the middle of this molecule. There's number 2. I forget which 3 they are. What's that? Lysine, serine, alanine, if I remember correctly. Looks about right. If you've condensed your lysine side chain so that it shows CH2 in brackets and a 4, you can get away with that. That's fine. Then this time it's what? Serine, lysine, alanine. So it's the same 3, but in a different sequence. Hopefully you're okay with that. And then the last one was phenylalanine and aspartic acid at pH 7. So it's getting a little bit silly here. I've pointed my R groups downwards, probably just to save space because there was something above. Doesn't matter which direction they're pointing. The only thing that might bother people is if it's pH 7 and it is an amide like this or a peptide, do you protonate this group? And the answer is no, but don't worry, you're not supposed to know that. So if you did, I forgive you. In other words, if you put an extra hydrogen on there and gave it a plus, okay? I had to look that up before I recorded this video just to check. But it turns out that when the NH is present in the amide functional group, it's not as easily protonated. It will not be protonated at pH 7. Like I said, you don't need to know that, so don't worry about it. Just a complication of the question that I created. Okay. So you can also make amides by acylation reactions. So this comes from the carbonyl topic. And so we already know this basically, we're retreading old ground. Amines will react with the acylating agents, acid chlorides or acid anhydrides. And the mechanism that follows is one that you are supposed to know. You didn't need a mechanism for the condensation reaction, but you do need this mechanism for acylation. It is nucleophilic addition elimination. So it's one of the more complex ones that you learn. So what we're going to do is some practice. Here's an example. One of the same examples I definitely would have used when teaching acylation. It is ethanoyl chloride reacting with ethylamine. And what you've got is the setup. So on the left, we have ethylamine next to ethanoyl chloride. You need to draw the appropriate curly arrows on that left-hand side. We then follow one large arrow to the right, and we have the intermediate, which also needs curly arrows on it. I could be nice and tell you how many, but I'm not going to. So we've got curly arrows on the left, curly arrows on the right, and then after we go downwards, we'll have a look at the products. So pause the video, copy these structures, and mark on the appropriate curly arrows as far as you can remember. Okay, so what we should have is nucleophilic attack on this carbon by the lone pair that's present on the N in ethylamine. Movement of the bonding pair, or one of the bonding pairs, in the carbonyl double bond onto the oxygen. That's 2 curly arrows to the left-hand side. We then need curly arrows on the right-hand side. Chlorine is our leaving group. It's going to be kicked out as a chloride ion. We're going to reform the double bond at the same time by moving that pair of electrons back that way. And we're also going to lose a hydrogen ion, which means the bonding pair that's holding the N and the H together moves onto the N. It does not matter whether it's the hydrogen on the left or the right. I've just chosen the one on the left. Okay, it's not important whether your curly arrow is there. I might as well show you. If it's there, that's okay. Obviously, you can't have both, but if you did one of those or the other, then that's all right. Right then, you get the N-substituted amide, N-ethyl ethanamide, we've already talked about that a little bit, and hydrogen chloride as a byproduct. Okay. Whilst we're here, I suppose I should also point out that if you have another molecule of ethylamine in your mech that takes away this hydrogen, you can get away with that because ethylamine will act as a base. Okay. And that would obviously change the product to ethyl ammonium chloride instead of hydrogen chloride. Doesn't need to be there. You don't learn it as being there, but I'm saying if you did, then actually, you know, that would be completely fine. All right. I just don't advise it as a habit because it's complicated enough already. If you have no idea what I just said, ignore it. Okay. So here's your next task. I want you to do either two things or one thing. You either outline the mech and name the product, that's in green, or you just draw the product, that's in blue. So you don't have to do the mech basically with the acid anhydrides, okay? But you do have to try and draw the resultant amide. So pause the video, draw out your mechanisms where appropriate, draw out your structures and names where appropriate. Okay, so we should be done with that, or at least done as far as you can be. Okay, so I'm not outlining the mechanism. Previously, past me has not outlined the mechanism, which is okay, to be fair, because, you know, if you could do it in the example that we did to begin with, you can do it now. So all I've done is named the products for the first two. N-methyl propanamide from the first one, N-butyl ethanamide for the second one. If you're not sure about whether your mechanism's right, just go back and cross-reference it with the original one. Those 5 curly arrows should be in the same spot. So then, for number 3, ethanoic anhydride and phenylamine would make this amide. And for the fourth one, you would get that amide. Hopefully, you can kind of see the logic there. It's the same idea, but again, as usual, to sort of ramp up the challenge, it's just incorporating aromatic rings. Even though the rings don't do anything in the reaction, as soon as they're there, it has a tendency to make students panic a little bit because they think, oh, sugar, do I need to do some benzene chemistry? A lot of the time you don't. The benzene chemistry that you learn is actually quite limited in a lot of ways. Okay? Right. Last little bit. Since amides can be formed by condensation reactions, it stands to reason that they can also be hydrolyzed. This is the same logic that we use when we're describing the equilibrium mixture that's present in esters. So if you think back to the carbonyl topic, esters are formed by condensation reactions, and they can also be hydrolyzed. And we don't need to worry about the ins and outs of that right now, but they're kind of the opposite of each other is what I'm trying to get at. Condensation produces a small molecule byproduct, usually water. Hydrolysis, in this case, uses water to split things apart. In other words, this is going to reverse the reaction. So you can pause the video and copy these structures and then predict for me what it's going to look like below that arrow, because in reality, if you've been following along, you should be able to see the reasoning here. Okay, we're going to get our original molecules back, basically. So here we are. Adding water across the amide linkage is going to get you a carboxylic acid back, and it's going to get you an amine back. Now, in reality, the reaction might be a little more complex than that, but that will do, right? For the purposes of this video, that will do. I think that takes us to the end. Yep. Okay. We are done. There we go.